And welcome to the inaugural webinar in the Mid-Atlantic Better Composting School webinar series. I'm Greg Evanylo, a professor and extension specialist in the Department of Crop and Soil Environmental Sciences at Virginia Tech. And I'd also like to introduce to you my colleague, Dr. Gary Felton from the University of Maryland, who has collaborated with me in developing this webinar series. So Gary, if you want to say hi.
Everybody, as Greg is, I'm also an extension professor. I'm at the University of Maryland. I've been doing compost teaching for about 20 years, and most of the things I've done, Greg has been my partner in it. We have the Better Composting School together. I work on poultry mortality, large animal mortality, do some research in compost use in turf, and on compost blankets for erosion control. Back to you, Greg.
Great. Thanks, Gary. So as a little bit of background to this webinar series, Gary revived what had been the Maryland Better Composting School about 10 years ago into a regional composting school, about a week-long school held usually in Beltsville, Maryland. And we've been conducting that school with help from regional and local instructors almost every year for about the last 10 years. We've also decided this year to do the school in the form of a webinar, which could be recorded and then kept online for posterity, so attendees can go back and review the information. So this composting series should be useful for those who are studying to take certification exams, simply considering starting a composting facility, for regulators who are interested in knowing more regular in the public who has an interest in composting. So for today's webinar, again, I would like to repeat that if you put any of the questions that you have in the Q&A box, Gary will be monitoring that during the seminar, as I won't be able to see them, and at various times during the webinar, possibly in the middle and certainly at the end, we'll stop and try to answer your questions. I'd like to also mention that at the conclusion of this webinar, in the chat box, we'll post a PDF of the notes for this webinar, and those notes are also posted on the eXtension Learn site that you may have encountered during your registration process. Also at the end, I'll provide a URL in the chat box for a very brief Qualtrics survey so that you might help us improve the seminar for future, or improve the webinar for the future presentations. And again, remember that this is the first in a series of 12. They're going to be monthly webinars on the third Friday of every month at 2 p.m. Eastern Time. So without any further ado, let's get started into today's webinar, which is the science of composting. And in this webinar, I'd just like to provide some of the science background that will later in future webinars be more, there will be more detail in the various management practices and testing of compost. So first of all, let's start off with what is the composting process. And here in front of you, so let's start off again with the composting process. And here in front of you, you see our typical composting pile. And what is it then that will be part of this pile that will result in our compost? And so we have a number of items to consider. Of course, we have our feedstocks, which we'll talk about. We have the microorganisms, which perform the microbial activity that decomposes the feedstocks into the compost. We have such important parameters as oxygen and water that are required by the microorganisms. And after they go through the compost system, the way we manage it, we have a finished compost that we will develop. In addition, we will during the process, we have generation of water when water vapor as heat is generated. We have CO2, carbon dioxide, that's given off as oxygen is converted by microbes to carbon dioxide, and we have heat generated. And the critical aspect of the composting process is this heat that's generated and retained in the pile to increase the temperature to select for the correct microorganisms that will efficiently produce our compost. So let's first of all talk about the final aspect of what could be produced in a compost pile, which we really don't want, and much of the rest of our webinar series will speak about this, are the potential and nuisance odors that might be generated. So let's look a little bit in general about how composting happens. Why does it happen? So again, microbes, which are ubiquitous in soils and organic matter, consume feedstocks to obtain their energy and nutrients in the same way we as humans or cattle will consume food and generate waste and convert it to protein and new cells also. The activity of these microbes, their respiration, creates heat, and then the heat gets trapped in the pile, and that accelerates the process. So remember that composting is a degradation process, but because we're managing it, and we can accelerate the process. And our management of composting is essentially a way to optimize the conditions for the microbes to do their decomposition. So let's look at the types of microorganisms that are active in composting. Bacteria are the most ubiquitous of these. They can be found as single-cell rod or bacilli bacteria, sphere or cocci bacteria, or spiral or spirilla bacteria. And they comprise about 80 to 90% of all of the organisms in compost. Bacteria are the earliest and fastest decomposers in the pile. And the reason is that they are most nutritionally diverse. They can subsist in compost pile with a wide range of nutrients. They are very tolerant of low oxygen in the composting pile, which is one of the reasons why they are one of the first decomposers, because during the composting process, as soon as the pile is formed, we typically create conditions of low oxygen almost immediately as the very active carbon begins to be broken down. Now we have different types of bacteria. We have those that are called mesophilic, or those which are those that survive at the medium temperatures that we most often know in our environment, the temperatures that we are encountering today outside of our offices. The thermophilic microorganisms, which I'll speak about in more detail in a bit when I show some temperature responses, are those bacteria that can survive at higher temperatures, and they're the ones that are quite active in the composting process. They are often during our normal mesophilic temperatures dormant, surviving unfavorable conditions by forming spores, sometimes called endospores, that are resistant to heat, cold, dryness, or lack of food. A second group of microbes in composting are the actinomycetes. These are classified as bacteria or similar to bacteria, but they do resemble and function a bit like fungi, so they are somewhere in between. You may know actinomycetes by the earthy soil smell that they have when you take a spade or a pitchfork and you turn over some fresh soil in your gardens. That earthy soil smell is that of actinomycetes. So actinomycetes grade more complex organic compounds than do the bacteria. They will degrade compounds like cellulose and some lignin that are found in woody stems and newspaper, chitin, and they'll decompose proteins via the enzymes that they produce. They'll appear during the hotter thermophilic stages and also during the cooler curing phase that occurs after the thermophilic stage. They will form long thread-like branched filaments that look like gray spider webs, and you can see a picture of them. We can often, you may spot them within compost piles. And finally, we'd like to mention the fungi, which includes molds and yeasts. Fungi are more tolerant of low moisture and pH, but they're less tolerant of low oxygen than bacteria. That's one of the reasons why fungi are not early decomposers, but typically function during the latter stages of composting, maybe somewhat during the thermophilic phase, but also during the cooler mesophilic curing phase at the end. And the fungi are the most efficient of the woody product decomposers. So let's look at a summary of sort of the progression of carbon decomposition during composting. So first of all, we have the very easily decomposable sugars and starches. They're the first to be decomposed, typically by the mesophilic bacteria. They generate a lot of metabolic activity. They generate a lot of heat and carbon dioxide and result in a rapid rise in temperature early in the process. Later, the more recalcitrant proteins and fats are then decomposed by the thermophilic bacteria and actinomycetes, and they generate medium heat and carbon dioxide. And finally, the most resistant of the carbon compounds, cellulose and hemicellulose, are decomposed by actinomycetes and fungi, largely during the end of the thermophilic and during the mesophilic curing stage. So now that we have a little knowledge of the background of the microbes, let's look at the temperature responses that we would expect in a compost pile during as compost occurs. So in this graph, on the left column, we have temperature in degrees Fahrenheit, and on the right side of the graph, we have temperature, the same temperatures expressed in degrees Celsius. And across the bottom of the graph, we will have time. So as we've initially mixed the feedstock for the composting, the feedstock may first have a temperature like we might be experiencing outside today of maybe around 70 degrees, at least in some parts of the country. Within a short period of time, the activity of those mesophilic bacteria decomposing those starches and sugars will begin to cause a rise in temperature of the pile as the heat is being retained in this pile, which has great insulative capacity. So we refer to this zone below 105 degrees, as that is being the mesophilic zone, and the organisms that function below 105 degrees are the mesophiles or the mesophilic organisms. Now, as the temperature rises above 105 into a zone that we call thermophilic, we start to select for the organisms that are thermophiles, and these would include the thermophilic bacteria. So again, remember those dormant thermophilic bacteria which exist again in soils and organic matter and are largely ubiquitous in nature would begin to wake up, and then at that point they would begin to decompose the material as the temperature would again continue to rise into the well above 120, 130, 140 degrees. Now, as we rise to this temperature, maybe between 140 and 150 degrees, we are in what we would consider the ideal zone, temperature zone, for composting by thermophilic bacteria. But if the temperature was to rise beyond that, particularly above 160 degrees, we would likely begin to retard the growth of those thermophiles as they do not function that well. There are other less effective decomposers that are active beyond 160 degrees. So part of our management process during composting is to maintain the temperature in this thermophilic zone, but below about 150 degrees. We like largely to see between 135 and 150 degrees Fahrenheit. Now we see these little squiggles, these up and down lines during the thermophilic zone, and the drop in temperature is often caused by what would be a process, one of the management processes that we use to release heat. We can do that either by blowing air into the pile or by turning the pile and releasing heat. And we'll talk about that in more detail later during the webinar series when we talk about the actual management processes. But for now, we use these various management processes to keep the temperature in the thermophilic zone, but not greatly above 150 degrees. Now we'll see that as the temperature drops, maybe through a turning of the pile and release of heat, shortly after that pile is reformed, the temperature will again rise, but it rises to typically a height or a temperature not as high as the previous temperature, and that's because there is less available decomposable active carbon for the microbes, and so on and so forth. So as the temperatures, as the microbes decompose the active carbon and the temperature drops and then rises, it never rises up again fully as high as earlier in the process, because our active carbon used by the microbes is being depleted, and there is less source of energy for their respiration. So we go through this thermophilic stage, and at some point, most of the decomposable carbon is depleted, and we begin to see a drop in temperature. And eventually, we drop that temperature back into another mesophilic stage, which we would consider to be the curing and maturation stage. During this stage, we expect that almost all the decomposable carbon has been stabilized, and we are now there are now other processes that are taking place. But for all intents and purposes, the largely active composting has been completed by the time we get to curing and maturation.
Got a question on temperatures, Greg.
Okay.
Is it necessary to achieve temperatures greater than 160 degrees to kill certain harmful bacteria or unwanted seeds?
Yes, and that's a good question, one which I'll address with a graph a little later during the presentation. But since we have the question now, let's address it now. So above 131 degrees, Fahrenheit or 55 degrees Celsius is the magic temperature for destruction of pathogens. So there's a time-temperature relationship. At that temperature, above 55 degrees Celsius or 131 C, for a minimum of 3 days, we can be assured that pathogens, both plant and animal pathogens, will be destroyed. At a little higher temperature, maybe 140 degrees, we can be assured that just about any weed seed will be killed. So we don't need to go to 160 degrees. In fact, it's somewhat detrimental to the process to go that high. Just getting above 140 to 145 degrees will largely kill every pathogen and every weed seed that one needs to kill in the process. Any other questions, Gary?
Nope, that's it.
Okay. All right. So thanks for that question. It's fine to try to answer them as they come along, since I think we'll have plenty of time today.
We got one question as soon as I opened my mouth.
Okay.
How does the weather affect pile temperature?
Yeah. Again, another good question, and one which I'll talk a little bit later about pile size and shape. But since we're talking about temperature and just ran through the composting process, let me again address it here, because many of these concepts you probably can't hear too often. So the composting temperature and heat is a biological microbial process, not so much of a chemical or physical process. And the microbes in these piles are very efficient producers of heat. And if your pile is large enough, and this is a critical point, that your pile needs to be large enough to provide the proper insulation, that the heat generated by the microbes will be very slow to leave that highly insulated pile. So we see composting occur in the middle of winter in Minneapolis, Minnesota, in piles that are ample size, because the microbial activity is generating enough heat, and the size of the pile is producing enough insulation to keep that pile warm. As your piles decrease in size, certainly the gradient of temperature from the inside pile to a colder outside temperature can affect the temperature in the pile. But as long as you're working with adequate size piles, outside temperature will rarely have any effect on the temperature in the pile. All right.
One more question, and this will be the last temperature question.
Okay.
Are there chemicals that could be part of the feedstock that might inhibit proper microbial activity?
So it is possible that if there were large amounts of fungicides or something else that was highly toxic, it could inhibit microbial activity. But with the types of products that we normally compost, animal manures that may be treated with various antimicrobials or biosolids or paper mill sludges, yard waste, we do not see such occurrences. Okay?
Okay.
All right. So good. So we got some initial questions. Hopefully we've done a fairly good introduction to the initial temperature increase and how the temperature varies throughout the process. Now let's look at the various process variables and how each of them may affect the process. So the initial feedstock properties, moisture, aeration, shape and size of the pile temperature, and compost retention time. So let's first of all look at feedstock properties. And when we talk about feedstock properties, we talk about nutrients. We always seem to talk about carbon and nitrogen, even though all the nutrients in plants, nearly all the nutrients in plants, are also essential nutrients for microbes. So why is it then that although microbes need phosphorus, potassium, manganese, magnesium, and so on and so forth, that we tend to focus on carbon and nitrogen? And the reason for this is that our carbon and nitrogen feedstocks, those are the materials that we mix together to form our composting pile, typically will not be limiting in any of the other essential nutrients. But in fact, they may be limiting in carbon or nitrogen. And again, to a reminder from earlier on in this presentation, our carbon provides the energy source, just like it does for us. Breaking carbon bonds provides our energy, and the nitrogen in these feedstocks, like for us, provides amino acids, which are building blocks of protein and building blocks of new cells. So in this picture, we see what are two feedstocks: a biosolids material, which woody waste that we would then mix together. But remember that all feedstocks will contain both carbon and nitrogen, and so whether they're considered largely a carbon source or a nitrogen source depends largely on its carbon to nitrogen ratio. So what is the carbon to nitrogen ratio? It is the relative amounts of carbon in mass to nitrogen in the mass of the mixed feedstock. And the reason that's important is the microbes need an appropriate balance of carbon to nitrogen. They need an ample amount of carbon for their energy, but then they need an appropriate amount of accompanying nitrogen so that they can continue to build new bug bodies and continue to produce more microbes to keep the process going. An ideal starting range of carbon to nitrogen is somewhere between 25:1 to 35:1. If you have what we would consider a high carbon to nitrogen ratio or a wide carbon to nitrogen ratio, for example, a ratio of greater than 40:1, that can slow the composting process. So there would be an ample amount of energy providing carbon, but there would be a very small amount of nitrogen, so there would be little growth of the microbes after the initial flux of carbon and after the initial production of heat. At the other end, having a low carbon nitrogen ratio, for example, of 20:1 or less, would not actually slow down the composting process, but would result in an excess amount of nitrogen per carbon providing energy, and this might result in some loss of nitrogen. So if one of the reasons you want to compost is to conserve nitrogen in the pile to make your finished compost a good source of nitrogen for plants, then selecting a very low carbon to nitrogen ratio would result in some of that loss of nitrogen and perhaps not make as good use of the nitrogen in your pile. So let's look now at some of the effects of the carbon-nitrogen ratio on composting. In this graph, we're showing the change in temperature, and an increase in temperature would show that composting is progressing well with days of decomposition. For a carbon feedstock recipe containing different carbon to nitrogen ratios. So here we have a material being composted containing a very high carbon and nitrogen ratio of about 60:1. This might be just hardwood tree leaves that are ground and put in a pile. So there would be an initial increase in temperature as the carbon is broken down, but the nitrogen in that nitrogen limiting source would be quickly depleted. The temperature would not rise high and it would fall quickly. Having a product with a lower carbon nitrogen ratio, maybe those leaves mixed with a small amount of yard trimmings, would result in a higher spike in temperature, but again, possibly not the most ideal composting, as it might still run out of nitrogen. But then here we have an ideal carbon nitrogen ratio of about 30:1. We see the temperature rises earlier, stays higher for longer before depleting. And this kind of a temperature profile is indicative of a composting system that has had a significant amount of decomposition. Let's move to the effects of particle size and shape on the composting process. Remember that the decomposition, that is where the microbes are feeding, happens on the surface of the feedstock. So smaller particles have more surface area per mass or per volume. So the smaller you can grind the particles, the more active food you can provide for the microorganisms. However, there is a limit below which you grind those particles that can limit composting, and that's if those particles were ground so finely that they compact together very closely and prevent the needed airflow to provide oxygen. So having particles too small can reduce the composting efficiency. And then we'll often add rigid particles that aren't meant to degrade, but really to provide structure in a pile to help for the aeration. So here we have a slide, and we've adapted this from a friend of ours, Tom Richard, a longtime compost educator, and showing different types of particle size and how they might affect aeration. Here in the upper left, we show a loosely packed, well-structured pile where we have materials of different shapes and plenty of room for air to move through. We can also have a loosely packed configuration, but where the particle size is somewhat uniform, and composting will likely perform very well here until maybe the particles become compact. And down here in the lower left, we see that the particles, in fact, are becoming compact and maybe preventing the flow of oxygen as ideally, and reducing the composting efficiency. We can also have particles of different sizes, which we would like, but we can have very small particles that are tightly packed within larger particles that might also reduce oxygen flow. So we must have a balance between the small size of particles that microbes like to eat and the large enough size particles for which they can provide oxygen. And here we have another slide showing the effects on particle size on decomposition rate. Again, temperature is an indicator of how well composting is proceeding by days of decomposition. Very large particles, 6 inches in diameter, we wouldn't recommend trying to compost these. These could be shredded wood, large wood chips, which might have, such a pile might have an initial temperature spike, not raising very high, and then leveling off immediately. But as we decrease the size of the particles passing through a screen, first to 1 inch, 2 inches, and then to 1 inch, we again see composting temperature spikes faster and stays higher for longer at the smaller particle size. Let's move now on to another parameter that composters will use in their operations, and that is bulk density. So bulk density is simply a mass, or on Earth, our weight per unit volume. And this can be expressed in pounds per cubic foot, tons per cubic yard, or in the metric system, kilograms per liter. Some examples of this are a cubic foot of water weighs 62 pounds, a cubic yard would weigh 1.44 tons. Dry topsoil, likewise, will weigh about 75 pounds per cubic foot, or 1.1 tons per cubic yard. And compost weighs about 44 pounds per cubic foot, or about 1,200 pounds per cubic yard. We use bulk density to be able to make conversions between mass and volume, because the composition of the feedstock is typically analyzed and presented in mass units. But when we actually generate or mix our piles, we're usually mixing them on a volume basis. So it's important to know the bulk density in order to be able to make the conversions of how many cubic yards of various feedstocks one would want to blend in order to provide the appropriate recipe, which has been determined by our recipes on a mass basis. And again, remember, lower bulk density means more porosity because the material's not packed as tightly, and more free air space. So too high a bulk density, again, will result in the particles being compacted too high and not favorable to good airflow. So let's look on now at pile moisture. And moisture, or water, is required by microbes, as it is for all life on Earth, or most life on Earth. And it's required for heating and cooling, and provides a little zone in the compost place for them to live. Now, the optimum amount of moisture in a compost pile is between 45% and 60% moisture. And that means the amount of water comprising the pile is 45 to 60%. So in a pile that would weigh 100 tons, for example, 45 to 60 tons of that would be water, and 40 to 55 tons of that would actually be solid mass. Okay. Greater than 65% moisture means the pore spaces are filled with water. So we can get what we call anaerobic conditions, that is lack of oxygen, and the microbes would therefore be limited in the amount of oxygen that they need for good composting. At the lower end, that is less than about 40% moisture, the piles become too dry for bacteria and actinomycetes. Some fungus can dominate at that temperature. There can be some decomposition, but typically you're not going to have much decomposition at less than 40% moisture. Also, once we allow the moisture to drop to this moisture content, we somehow need to re-wet the piles. And re-wetting compost piles, as we'll see in some of our later webinars, can be very difficult. Below about 35%, you face a great dust problem where much of the compost small particles can be blown around the site. So here we see some typical cross-section in a compost pile with different particles. With the ideal amount of moisture would form a thin film around the decomposing particles where the microorganisms are decomposing, and then we would have larger air spaces or porosity for airspace to bring oxygen into the pile and to move carbon dioxide out of the pile. Did I hear a question there, or? There
is one here.
Okay.
If the moisture gets above 65%, is there an odor problem?
So there can be an odor problem because the there are anaerobic microorganisms that will cause fermentation which is decomposition of carbon without oxygen, rather than composting, which is an aerobic process. So that is one way to know that your pile may be too wet, as you can smell the effects of anaerobic decomposition products, acetic acid and butyric acid, and some reduced sulfur and nitrogen compounds.
It smells like a septic tank.
All right. Is that the only question there, Gary?
That's the only question. Go ahead.
All right. Okay, let's move on. So, so as we would move, you know, we hear about oxygen, and there was a good question about oxygen and moisture, and so this is one of our key aspects besides moisture of one of our parameters that is very important to manage correctly. So aeration, somehow, usually either blowing air into the pile or briefly turning the pile, will supply some oxygen. We know that oxygen consumption will increase with temperature, as the temperature is a result of the respiration by the microbes during their converting of carbon to carbon dioxide. It's important to know that bacteria will actually start anaerobic respiration, that is decomposing under anaerobic conditions, when oxygen in the pile gets as low as 10%. And this will result not only in the odors, as we mentioned a few minutes ago, but it'll result in a slowing of the decomposition. In this little sidebar, we see that we want no less, and hopefully greatly more than 5% oxygen in the compost pile for aerobic decomposition. And one might wonder how difficult that will be with about 20 or 21% oxygen in our atmosphere. So I'll use a graphic to show how quickly oxygen can be depleted in a composting process. But first, let's just look at the normal process of oxygenation in the compost pile. So here we have a compost pile. If it's not being turned or air is blowing through it, we get most of our oxygenation taking place through convection. So that is, as the heat in the center of the pile begins to rise, the warm air rises up the center of the pile, creating a little bit of a vacuum and causing some cooler ambient air to flow in from the outside. So that's one way that the pile naturally will oxygenate itself. Unfortunately, we can't do it efficiently only based on this sort of static method of aeration. Other methods that are used is, for example, forced aeration, where we can actually blow air into a pile. One of our later speakers will speak about this in the webinar on composting systems. So let's, though, look at a system that's been oxygenated by one of these forced air blowers. And this is a system that uses sewage sludge plus sawdust in what we call this aerated static pile. So this is one of these systems, as you've seen in the previous slide, where the feedstock is combined and then placed on perforated pipes above the pipes, through which oxygen can be blown into the pile from one of these blowers. And in this figure, this graph, what we're showing is how the percentage oxygen changes in the pile with time. So we start off at the formation of the pile. The oxygen in the pile is almost 20% near ambient outside at time 0. The pile, the feedstock is formed at this, and if we're freshly developed feedstock, we see that within 20 minutes, the oxygen concentration in this pile of very fresh material having a high oxygen demand will drop from about 18% down to maybe 2 or 3%. So remember, we'd like to see the oxygen maintained up in this area, and for most of the first 20 minutes, the oxygen is operating probably in an undesirable area. These aerated static pile composting systems typically operate where a blower will be turned on every at regular intervals, and this blower was designed to turn on every 38 minutes. So we were operating this composting at this low 2 or 3% oxygen for another 18 minutes. So at the end of 38 minutes, most of the oxygen or most of the aeration in the pile was less than desirable. At this point, the blower was turned on and the oxygen immediately rose. So this shows why it's important to have either oxygen blowing through the pile regularly, and also why the turning of a pile by a windrow turner, which may turn a pile and oxygenate it once every few days, will not likely provide the ideal oxygen situation or conditions in the pile for the ideal decomposition of the microbes. And again, another figure to show the effects of turning frequency on composting, and this would be maybe a windrow turner where the piles are turned either every 30 days, every 10 days, or every 3 days. And as we go to more frequent turning of the piles, the temperature rise faster and they stay higher for longer, resulting in better and even decomposition. So the turning frequency both affects the aeration, but also it will affect the particle size as the compost turner may beat up and continue to turn and homogenize the particles in the pile, and also fluffing them up for continued good composting. Now let's move on to pile shape and size. And there was a question before about the temperature. Will the colder temperatures outside or hotter temperatures outside affect the pile? And largely it's going to be the composition of the feedstock and the size of the pile that will affect it. So smaller piles allow for greater airflows. So if you build smaller piles, you can get more natural airflow to the center of the pile. Larger piles will retain temperatures, higher temperatures, but excessively large piles will increase compaction, as the large mass of the pile can press down on the particles within the pile. You can have compost with a somewhat large pile if you have good structure in a pile. So that is if you have particle size of varying distribution, including some larger particles like wood chips that are designed to provide porosity rather than a carbon source. You can have a bigger pile if you have a higher carbon to nitrogen ratio. So if your oxygen supply drops a bit, it's less likely to produce the noxious, not decomposed nitrogen odors. And you can have a higher pile, a bigger pile, if you have a fluffier pile, lower bulk density. Again, we will talk about constructing the pile and how big the pile should be in subsequent webinars when we talk about systems. But in general, I'll just say that your pile size is usually determined by the kind of composting system that you're using and the equipment you have to turn your pile or to build your pile. Here we have the effects on pile size or volume of pile on composting rate using a 20-gallon can, 15 cubic yards, and 25, and these should be cubic yards, not square yards, but 20-gallon can, 15 cubic yards, and 25 cubic yards as an example. 25 cubic yards is about or 25 cubic feet is about 1 cubic yard, and 1 cubic yard is about the minimum size that one should compost if one wants to properly insulate the pile and hold in the heat and maintain the high temperature. So we see as we go to smaller size piles, or in this case a can, that composting doesn't function as well, and it does so fairly well at a minimum of 1 cubic yard, or about 27 cubic feet. Another point to be made from this slide is there are many home composting systems that are sold that are very much smaller than a cubic yard. They may be the size of a 20-gallon can, and you can see from this graphic that one would not expect very good composting to occur in those kind of systems because of the lack of mass and ability to insulate and hold the temperature and hold the temperature at the high 131 degrees plus. Now we'll move on to temperature. Just check with Gary to see if there's any questions on the last few slides we've talked about.
We're good.
Okay. Great. So let's go to temperature, because this is the driver of the process. The first thing we'll say about temperature is higher temperatures lead to faster decomposition, but excessively high temperatures over 160 degrees Fahrenheit or 71 degrees Celsius reduces microbial diversity and slows composting. Most weeds and pathogens are killed at temperatures of 131 degrees Fahrenheit or 55 degrees Celsius or higher. So here's this figure showing again the relationship between temperature and respiration. The blue squiggles are the amount of carbon dioxide that is being given off by the decomposing microorganisms, and we could see that our temperature curve smoothly follows the activity of the microorganisms, again showing that our temperature response is directly related to the respiration of the microorganisms. So here we have a cross-section of a windrow with the likely temperatures that one might encounter. On the outside, these hashed lines here are less than 155 degrees Celsius, which again, remember, is less than ideal for composting. This area, represented by these hash marks, is the ideal temperature range of 55 to 65 degrees Celsius. Then we have an area inside a little higher where composting occurs. And this area here should be represented by these hash lines, are a little higher in temperature and an area zone of excessive temperature. So much of the center of the pile is functioning at the appropriate temperature, but the outside is not. And so we'll talk in future webinars again on various systems for ensuring that the outside material can be worked into the inside of the pile or be insulated in a way as we can maintain our necessary temperatures here. I'd like to bring up a concept called PFRP, or processes to further reduce pathogens. This is the process that we use a combination of time and temperature to ensure that pathogens are killed. Again, in aerated static pile and in vessel systems, we rely on 131 degrees or 55 degrees Celsius or higher for 3 days. In the turned windrow system, the same temperature, but we typically require longer days with more turnings to ensure that all parts of the pile will be subject to that higher temperature. And so if you remember our initial temperature diagram, where we go from mesophilic to thermophilic, here we have our PFRP of 131, and this is the area that we want to have our temperatures above 131 degrees for the appropriate amount of time to kill pathogens. So in summary of the various parameters, just our initial carbon to nitrogen ratio, again, between 25 to 1 and 35 or 40 to 1. Our moisture content in the pile should be ideally between 50 and 60%. Oxygen concentration at least 5%, but hopefully greater than 10%. And the temperature we want to maintain in the pile is ideally between 55 and 65 degrees Celsius, or between 131 and 149 degrees Fahrenheit. Initial bulk density, that is mixing the various feedstock to achieve a bulk density of about 1,100 pounds per cubic yard. We would like to see a variation in distribution of particle size between about 1/8 and 1/2 inch in diameter. And pH is somewhat forgiving. We can have a pH in these materials as low as about 5.5 to about 8, and the microbes will still do just fine. The final parameter I'd like to talk about is time, and I'll do it by talking about the various phases that composting goes through. The mesophilic phase, depending on the system, can be a few days. That is, remember, the mesophilic is from ambient temperature to about 105 degrees Fahrenheit. It could be a few days to a couple weeks, depending on the system and the feedstock. The thermophilic decomposition, maybe 3 weeks to several months. And then we have a final stage where the temperature drops down, curing and maturation, and that could be 1 or up to a few months. And it's during this phase that various inhibitors to seed germination and crop growth might be eliminated. So let's mention curing. It's a mesophilic stage, temperatures back to ambient, outside temperatures. There's not much active carbon anymore for the microbes, so respiration is reduced. Oxygen uptake is reduced. There's not as much heat being generated. Most moisture that's lost has been lost. There's not be much moisture to be lost. But it is also during this stage that we get a recolonization by beneficial soil microbes that we'll talk about much more in the compost use and benefits section. This is a stage that we get lots of useful properties for compost as a great soil amendment. So the property changes we'll see at completion of composting. The temperatures are typically less than 10 degrees warmer than ambient temperatures. We shouldn't be able to recognize the original materials. Composting typically has a dark color. It's stable. It should smell earthy, not putrid. The volume of the pile might be reduced by about 50%, and that's due to compaction and loss of mass. And the mass can be lost, might be lost as much as 1/2 or 3/4 of the pile, also due to loss of dry matter, but also loss of moisture during the heating process. So with that, I'll end up providing a few composting references. This on-farm composting handbook has been sort of the Bible of composters. There's a good biosolids composting handbook put out by the Water Environment Federation, and Haag is has produced a great engineering handbook on composting. So with that, Gary, I guess
got a few more questions,
and we can take as many questions as we have time in the remainder of the period.
Okay, the first question we got was about PFRP. Could you repeat the number of days to kill pathogens with respect to the type of pile, and I think also needs time and duration.
Yes, sure. So
temperature and duration.
Right. So at 131 degrees or higher, or 55 degrees or higher for 3 days, all pathogens will be destroyed. The reason that we have different time requirements for the system is because in different systems, we may not expose all the composting feedstock to that same higher temperature. So in the aerated static pile, which will be talked about under systems, or in an in-vessel system, all of the material in the pile is subject to that high temperature, and so 3 days is what's necessary. For composting in windrow system, where you would have some of the feedstock sitting on the outside of the pile, where the heat would be moving to the ambient outside temperature, we recommend, and number of groups, including the Organic Farming Association for compost to be used there, recommends 15 days of composting with 5 turnings. So that's a turning at least every 3 days. The piles are typically turned earlier. But as long as all parts of the compost pile can be at 131 degrees for at least 3 days, you should be able to meet PFRP.
Next question is, what are the inhibitors of germination that are eliminated during curing?
Mm-hmm. So even though our composting process is supposed to be done under aerobic conditions, we will often get the formation within small little pockets of the pile of the products of fermentation rather than the products of composting. And some of these products of fermentation are volatile organic acids, such as acetic acid, which is vinegar. And those of you who've ever had a pile of compost or a pile of mulch, particularly, delivered to your house, and you can smell the vinegar coming off of it, it's an indicator that some of the decomposition occurred under the lack of oxygen. And therefore, instead of only carbon dioxide being formed as the end product of decomposition, you actually had acetic acid being formed. There are other of these volatile organic acids, butyric acid and lactic acid, and these kinds of products, when they're applied to directly into the soil, or for where seeds are planted, or may be applied over near some turf grass or within a seed bed, can actually be phytotoxic to seed germination or to seedling growth. And we'll talk about this and how to measure this in much greater detail in our webinar on compost quality assessment.
One last question so far. Can having woody material, such as wood chips, keep temperatures higher for a longer period of time and extend the time it takes before temperatures drop to curing levels?
So the value of wood chips in the pile, because they have a relatively small surface area to their volume or mass, is mostly that of being a bulking agent. So the microbes themselves can feed off the surface, but if you were to take a wood chip and grind it into smaller particles, there would be much more active carbon for the microbes. So in fact, what the wood chip does in the composting process is to provide structure and aeration. And by doing so, it may in fact extend the composting of the pile, but it would have to do that through the continued aeration of the pile, because as soon as the microbes sort of feed on and the surface of that wood chip and stabilize that wood chip, it will no longer really be an active source of a lot of carbon. So it's, rather than putting a lot of wood chips in, it's more important to design your recipe to have a good mixture of fine and more coarse particles like wood chips. And as you'll see in some of the ensuing webinars, the wood chips then are typically removed, recycled, and maybe reused rather than rather than maintained in the finished compost. All right, any other questions?
There was also a question in chat about, Dominique says, what is the impact of including PLA items in your pile, e.g. compostable and takeout items?
Okay, so PLA, for those not familiar with the term, is the term for polylactic acid. It is a compound, an organic compound extracted typically from cornstarch to produce decomposable materials like decomposable cups and utensils and cups and bowls, and those have been demonstrated to compost just fine in a compost pile if you maintain the pile in such a way as to allow it to compost long enough. Sometimes they take a little bit longer than yard waste or some of our more natural products. And I want to also invite my colleague Gary to chime in on any of the answers if he has more insight, or sometimes to correct me.
No, I'm not. I'm too shy to do that. Got another question for you. If a county starts a brown bin collection, is the feedstock available sufficient to produce a correct C to N ratio, or do you need to seek other sources of feedstock?
Mm-hmm. So by a brown bin collection system, I'm going to assume that we're talking about woody waste, shrubbery trimmings, and hardwood leaf clippings. And if if you mean more than that, you can send something else to Gary in the chat board. But those products together, mixed together, will typically give a carbon-nitrogen ratio a little bit higher than what is desired. And it would be useful then to add a composting source that contains a more nitrogen in it, such as grass clippings. Now, we always recommend that grass clippings, when possible, are kept on the lawn rather than collected. But if for those municipalities that collect any lawn trimmings, they could be provided mixed with brown waste, typically at a ratio of about 1:3 or 1:4 to provide an ideal carbon-nitrogen ratio. The other alternative is just if you have enough space and you can compost long enough, you can compost yard waste and leaves and trimmings with a less than desirable carbon to nitrogen ratio just for a much more extended time period. That might mean a year or more, but it would be difficult to do it under the ideal conditions.
We're going to go the other way with this question. He's actually talking about brown bin collection system for table scraps.
Oh, okay. So now we're talking about food waste, which is at the other end of the spectrum. So that would be um, a much lower carbon to nitrogen ratio, typically below 20 to 1, and often having a very large high amount of moisture in them. So in that case, you would likely need the sort of brown products that I was talking about in my earlier answer to your question, which is more the woody waste, the yard waste in the form of tree leaves, shrubbery trimmings, and such. So the food products again are going to be quite low in carbon, nitrogen, high in moisture. If you try to compost them as is, you'll result in a lot of putrid odors. If, in fact, composting even can begin at the typically high moisture content of those products. So hopefully that was hopefully an adequate answer.
Yep.
In the spirit of no question is a dumb question, I'm actually I'm gonna put one out there. So I'm just a home composter, but I know temperature is important. Is there a good way to get the temperature of a compost pile? Is there a special thermometer or an infrared tool? What's the best way to get the temperature of a pile?
Yeah, good question. And again, we will have later webinars on instrumentation, but I'm not going to ignore your question now, Mark. There are, in fact, long-stemmed thermometers that are used commercially and can be used and purchased by the homeowner. They may be anywhere from 2 to 3 feet long. They will instantaneously or very quickly change temperature, and so one can monitor their temperature in a pile using those. While we're talking about the homeowner and temperature, I'll also mention that there's a way for a homeowner to estimate moisture in their piles too, if you don't have a problem with ickiness. And so we can have very nice scientific equipment to take a sample and dry it out and weigh it before and after to determine moisture content in the pile. But we also have a very nice moisture meter at the end of our arm, which is called our hand. And we can pretty much calibrate our hands to know about the ideal moisture content in a pile. It should feel damp, like a damp sponge, but you shouldn't be able, when squeezed, but you shouldn't be able to squeeze any moisture drops out. If you can squeeze moisture drops out of the mixed feedstock, it's likely too wet.
Yeah, I thought surely there's a thermometer. I was thinking to myself, I'm sure there's some long thermometer you can use, just sort of poke in there. So I'll do a search on long stem thermometer.
Yeah. Or join into our instrumentation webinar.
I'll be there.
Some more questions, Greg.
All right.
Is palm waste a good carbon source for compost?
So there's been some questions that I've seen on the U.S. Composting Council chat board, and for those of you who are not familiar with the U.S. Composting Council, it is the largest trade organization of composters in the world, and for anybody involved in composting, it would pay to become a member. And the chat board provides an opportunity also for providing questions and getting answers through the board. And I do recall some discussion recently about palm from an area of the country that I was not from, and from products that I'm not familiar with. But my recollection is that it can be composted. I don't recall off the top of my head what its carbon-nitrogen ratio is like, but it is a coarse kind of material. It may need to be ground, but certainly in the areas of the country where palm is produced, it is processed and used in composting piles, but I could not tell you anything about its carbon to nitrogen ratio without doing some homework.
I can jump in here a little bit. I taught a school at Apopka, Florida, which is north and west of Orlando. Anyway, they were getting parts of palm trees, if that's what you mean by palm waste, where the fronds and cut up pieces of wood were being sent in. That product was pretty difficult. It's very fibrous. It doesn't break apart easily. So they were having difficulties composting it, mostly because of its structure. So if you put some physical effort or mechanical effort into shredding it apart, it would probably compost fairly well. But because of its structure, it did not compost well.
Great. Thank you, Gary. Good. Like always, it's great to have you next door. Digitally. Digitally speaking.
I'm getting questions faster than we can get to them. So how does a home composter measure compost moisture? I think you just told them with the squeeze test.
So also, in addition to the squeeze test, if one wants to be a little more scientific, and one has a weighing balance or scale, you can weigh your wet material and then dry the material if one has an oven that dries as low as about 150 or 160 degrees Fahrenheit, and then weigh it again. And the difference between the weight of the wet material and the material after being dried in an oven at 150 or 160 degrees would give you your moisture content. But again, the squeeze test is one way that is much simpler and quicker.
Next question. How would the end products differ between a properly composted manure pile and a static pile, I assume of manure, that has sat untouched for a year or two?
So the static pile would have a rapid heating in the center, and then likely these sort of piles will often heat to a very high temperature, where they will often get so high it'll kill the microorganisms. And in fact, I believe, Gary, I'll let you chime in, but I believe Gary works much more with animal manure than I do. But there's the reason behind stacking of certain animal manures, like poultry manure, is to allow some high heat to be produced to kill microbes in the pile during storage. But I assume you would eventually get a little bit, as the temperature in the pile then cool down over time, there might be some reignition of composting, but it's going to be greatly reduced by the inability of air to move into the pile. The outside will decompose. We won't call it composting. It'll decompose the way tree leaves do in the forest floor. But so you'll have a very heterogeneous decomposition product from the outside to the inside of that pile, and so will also be the nutrients in there. You may, as you heat very highly in the center of the pile, volatilize and lose some of the valuable nitrogen products in there. And then unless the final product was mixed well together, the nutrient homogeneity throughout the pile will be lacking. Gary, you want to jump in there and anything to add?
In terms of usefulness as a final product, if manure piles sit around for a couple of years, They're not greatly that different, except that they have had the potential to leach some of the nutrients out. So you may have a lower nutrient content. It may have by that time gotten pretty moist, but it's still not a poor product. Another question: is the composting industry still concerned with persistent herbicides like glyphosate in finished compost?
So your question is partially correct. So the composting industry is, in fact, still concerned about persistent herbicides. For those who are not familiar with these products, around the turn of the century, there was a class of herbicides known as pyridine carboxylic acids, which act as plant hormones, and so they were used in largely in herbicides that was applied to grass. So for pastures and haylands, a product called picloram, which I believe is still widely used, contains some of those products. And in turf grass, a product called clopyralid and other similar products was used. The composting industry found out about these, unfortunately, when feedstock from these either turf grass treated with these clopyralid compounds, or when feedstock, or when hay from a pasture was fed to an animal, and then that animal's manure was composted. And even though composting will decompose these compounds, maybe as much as 95%, because they act as hormones and not as poisons, direct poisons as other herbicides do, they are bioactive at very low concentrations. And so the application of these composts onto plants of broadleaf types of plants, particularly tomatoes and snap beans and such, resulted in the death of many of these plants. Over the years, the industry has decided to, that is the herbicide industry, has cooperated with the composting industry to only permit the use of these compounds or these herbicides in situations where there is someone with a certain category license. They couldn't be purchased by a normal homeowner, and that label also included the statement that these materials shouldn't be used on products that would be composted. Now, while glyphosate also acts as a sort of hormonal kind of activity, it does degrade thoroughly and quickly, and has never been shown to be a problem in compost. Plus, one would never compost something that has received glyphosate, because glyphosate will kill it all. It won't. It kills. It's a full spectrum herbicide, and you won't really have any biomass after you've applied the glyphosate.
Here's an interesting one. Can you compare the results of composting to accelerated thermophilic aerobic digesters, ATAD? Mm-hmm. When you hear companies say they make compost or fertilizer pellets in 5 days, are the properties of the two products the same?
So ATAD is an accelerated aerobic thermophilic system. It's one that I'm not. I don't know a lot of details about. If it's truly aerobic and not anaerobic, such as we do have anaerobic digesters that some people would erroneously call anaerobic composting. So anaerobic digesters are not composting. They create methane. They create these byproducts, these volatile organic acids. I'm not familiar with the end products of the ATADs to know exactly, or these all the details of the process to know whether the process is in fact fully aerobic and results in a stable aerobically decomposed process. Gary, is there anything
I wish I knew more about that.
Yeah.
There's a second part to this question.
So also, let me, before you get to the second part, I'll mention that in the systems area, we'll have Craig Coker will speak, and Craig has some more knowledge of these kind of systems. So we could get an answer for that in a subsequent webinar.
And you know we're going to do a little research between now and then.
Yep.
The second part is our county is over 75% ag land. Which product would be better? Maybe we should postpone that one until we can get a better answer.
Which product would be better? Do we get a choice?
Compost or the ATAD product?
Oh, okay. Again, yes. So let me answer that, or make an attempt at answering that without knowing all the details about the ATAD system. When we produce a compost product, we are tying up much of the plant available nitrogen and reducing plant available N. So many of the benefits of compost as a soil amendment accrue from its organic matter content, some of its disease antagonism. It may be a biostimulant under drought conditions. So and it can help improve soil physical and chemical properties. Whereas some of these other design systems like anaerobic digestion and some forms of aerobic digestion decompose the product but conserve more of the nitrogen, and more of the nitrogen is in a more plant available source, or form. So it's possible, this is what a farmer might often look at. We rarely see farmers willing to purchase compost because they are looking for more uncomposted manure or biosolids that has a higher nitrogen content, and compost is more often used by landscapers or organic farmers that need a compost product. So I'll leave it at that for now, just some fodder for future discussion.
Well, I was going to say, guys, just so you know, we'll have transcripts of all these questions and who asked them, so we can certainly follow up with an individual after the webinar.
I do see that it's getting to be around 3:30. I guess we're running longer than we had anticipated. I'm not sure what time. We hadn't made plans to figure out exactly when to stop.
I think we may start running into some limits with regard to file sizes for the recording, so I'd recommend.
I'd like to go quickly over two more questions.
Okay.
How important is covering a pile during curing?
More important than covering a pile during active composting, because there's less heat being generated, and any rainfall or moisture that might permeate into the pile is more likely to cause some damage and not be turned back into water vapor. Okay.
Does biosolid compost need a special notification to customers?
That might be site or state specific. So in Virginia, it doesn't require a specific notification, but the only thing where biosolids compost is different in the marketing arena that I'm familiar with is in the organic market. So right now, if you're a certified organic farmer, you cannot use any biosolids-based products, including compost?
In Maryland, biosolid products have to be tracked and reported back to MDE as to where they went on, but they're not reported to the public.
All right. So, Gary, I'm thinking with that, we could probably call it an afternoon. I'm thrilled of all of the interest in at least the first one of these. As I mentioned that hopefully Mark is putting a URL for the PDF notes in the chat section.
I've done that, and if you guys go to chat, you'll see a link for the learning event, so you can access the recording there.
Yeah, I don't have access to that to place the Qualtrics survey, so
so you can stop sharing if you want.
Okay, but let me also, I'll put a Qualtrics survey in the chat that I would appreciate you, those of you still online, to answering. We will, as we move to subsequent webinars, provide notification, as we will provide notification of the recording of this one.
And while you're putting that link in there, I guess you may have planned on doing it, but just a plug for the next webinar, July 21st. So mark your calendars, and I'm assuming maybe there's an email list that notifications will be sent out to. So, and the rest, or the title I have there for that July 21st webinar is Feedstocks and Recipe Development.
Mark, at this point, they've all lost the chat window. Is that correct?
No, no. Everyone still has access to chat.
Okay.
All right. So let's see, chat.
And that's going to be down, Gary, in the bottom of your Zoom window there.
I moved it.
All right, and I think I just. That's. How come my chat says to Gary Felton privately?
Right, so you'll need to change that to,
Yeah.
you want to make sure you do all panelists and attendees.
Very top.
If you click that little, if you click where it says to Gary, then you should get a drop down or a pop up.
Yeah, I'm not.
Okay. Yeah, I'm not sure why. Another option could be, Greg, you could share this via email. I don't. There's no way for me to see your screen, so I can't tell what you're seeing.
Oh wait, to everyone, to everyone, right?
Yeah,
panelists?
yours may say yours may say right. I think everyone will do it. Yeah.
Okay.
Yours is a little different than mine. Sorry.
So.
And you want to paste that link in and hit return.
Okay. So return on your
keyboard.
Yeah, I did.
Yeah, I'm
out?
not seeing it. No, I'm not seeing it, so I'm not sure.
Did you type the message in below where it says all panelists and attendees, or everyone?
Yeah, well, right where it says to.
Right below that. Mine says type message here.
Yeah, well, that's where I typed it. Let's see. I can talk about compost, but I oh, I think it might have happened.
Yeah, let's see.
Yeah,
everybody. Yes.
Yay! Thanks for bearing with me, folks.
Those of you that are still left, please go out and use that.
And yeah, Greg, we've got, if you just as a reminder, Greg, there's a Zoom has a mechanism for sort of making that process a little easier. So I'll try and remember that before the next one. But if I forget, you remind me about allowing people to enter the survey.
All right.
Okay, so I think we're good. Any closing thoughts, Greg or Gary?
Not I. Again, the next one, next session will be on feedstock and recipe development, and that one will be jointly done by Gary and me.
All right, folks, we will see you in 1 month then. Thank you.
Great.
Thanks, guys.
Mark, thank you so much.
Sure. Bye-bye.